Hello everyone, welcome to our knowledge series and today we are very excited to have two different regions joining hands to this virtual platform. So we have today Rich, which is Research and Innovation Circle of Hyderabad, represented by Mr. Ajit Rangnekar, who is our very kind supporter and always encouraging us to do some good work. And we also have Dr. Janusz Pikani representing Estonia, representing ScanBalt region. He's chairman of ScanBalt region. And as you know, Estonia part of EU and Telangana state of India. So both are doing some interesting work. Both regions are doing some interesting work, and today we are going to discuss and understand more about the challenges and opportunities from their point of view in terms of the collaboration. And just some housekeeping assignments, announcement here that I welcome you all to type questions as they come to you. But once our speakers today make their presentation, we'll take the questions toward the end of the webinar. So keep typing them, and we will take them towards the end of the webinar. So now I come to introducing our moderator, as I already mentioned about Mr. Ajit Rangnekar, who is the Director General of RICH. Uh, Mr. Ajit Rangnekar, has been very instrumental in, bringing together initiatives which were first of its kind. If I may say, Indian School of Business, a premier business school, is one such initiative which came, which saw light of the day in the city of Hyderabad, and now today is very well known all over the world. And now with his current assignment, Mr. Ajit Rangnekar is looking to foster research and innovation in the city of Hyderabad and the state of Telangana. And personally, my interaction with him has been very, very, what do you say, uh, positive. He has always been encouraging to do new things. He's a very kind and generous person. And I request you to take over from here and share with us what Telangana has to offer, what Hyderabad has to offer, what research Rich can do for us. Over to you.
Thank you, Sachin. Thank you. And thank you for the very kind introduction. I wish I deserve it, but I shall certainly from now on work towards it. And good afternoon, good morning, good evening to all of you, whichever, depending on whichever part of the world you are. I'm delighted to be a part of this webinar on evolving research in medicine. You know, nowadays when we talk of evolving research, the latest buzzword is all about precision medicine. And originally, the definition was more focused, like the word medicine is on treatment, but increasingly that word is now expanded really to call it precision health or precision medicine to cater to both disease prevention and health and treatment. And therefore takes into account something more than what we have done in the past, which is the individual variability. And a lot of that has come on account of our new research in genetics and subjects like that. It's also now, and certainly in India, been fostered by new diagnostics and digital devices, and of course, the big move in data and analytical tools. So, you know, these things would not have happened if all these advances in research and diagnostics and analytics had not happened. And therefore, this is a pretty exciting time for us to look at what the future of research in health sciences is going to be. Now, I've given at the bottom of this slide some numbers. And as always, with any number to do with anything in the future, take them with a pinch of salt or even a bucket of salt. All we can say is that this market is going to be big, and we are going to treat with huge amounts of data. Right? But there are some very basic and some very important considerations which we find were not there when we did our research in medical and life sciences earlier. The first is, how do we deal with the complexity of data, right? And this is especially true of clinical data, because clinical data, there are really no standards. You may have certain standards for certain medical tests, but certainly, how do we collect, how do we standardize, and how do we aggregate medical data and medical histories is going to be a big challenge. The second thing is, how do we derive insights from this? There is the science of genetics, but there is also clinical knowledge. And unless those two come together and work with data scientists, the insights are not going to be adequate. So it's going to require a fairly large collaborative effort between various communities. Now, in the past, if one said that your, fasting sugar was 140, then you kind of said, okay, therefore you're a diabetic. That is no longer going to be true, because each test will have to be looked at in the concept of each individual. And therefore, the same data may have different values to different people. So how do we actually figure that out? How do you get to those insights? Earlier, we used to do it for large numbers and therefore draw common conclusions from that. That will change. We haven't even begun to think of business models. And I think that's going to be, especially for people like me who come from the business background, that's going to be one big fertile field about who pays, who does, who owns the data, how is that data exploited, and so on and so forth. Again, for people like us in India, scalability is a big problem, as is the cost of these devices and therapies. And this is equally true of Africa. Large continent like Africa, or a large subcontinent like India, where you have over 1 billion people. How do you ensure that these technologies become scalable, become affordable, etc., etc.? And finally, the biggest elephant in the room: how do we regulate data, especially issues of data access, privacy, etc.? Fortunately, there are a few people like Dr. Janusz Pikani who have done a lot of thinking on this. So a little later, I'm going to ask him to cover some of these areas. Just to set the scene, in India, we are fairly early in terms of doing work in this area. And what's interesting is that India is really not one country genetically, unlike a China where maybe 90-95% of the population is Han population. In India, there are a large variety of population groups, further complicated by consanguineous marriages, inherited genetic orders. But there are a large number of institutes, research institutes, hospitals, and some even startups, some of whom we are working with, who are already beginning to do research in this. India's strength, of course, is that we have all the state-of-the-art facilities, we have outstanding physicians, we have a great pool of life sciences researchers, we are, of course, well known for our IT capabilities, we are a big market, but there are some challenges too, right? We don't, as I mentioned, we don't have data about the Indian population. And how do we create a database which represents 1.2 billion people? It's a big challenge. How much will it cost us? How will we be able to afford it? And where will we find the clinicians who will be required to adopt these things? So those are some fairly big challenges for us. For different people, there will be different challenges. We already saw one question from Spain which talked something similar, that how do we bridge this gap between the research and its bringing into the market? So it's not just one country that has it, it's probably widespread. And finally, we are looking at some stage, how do we set up genetic studies in India? How do we create standard operating procedures? How are we going to store these samples? Most importantly, we are all talking of human beings. How do we create partnerships between people who normally up to now have never really collaborated? And finally, of course, most importantly, how do we have a regulatory framework that protects an individual, at the same time allows and encourages research? So these are some of the challenges which I think are going to face us. Fortunately for us, Estonia has done some amazing work in this area. And we are today really happy that Dr. Janusz Pikani is with us. And I'm going to very quickly read out his CV to you. But before I do that, a word of caution. Some of you listening to it will think that I've mixed the CV of 5 or 6 people. I have not. This gentleman is really, truly an amazing person. So Dr. Pikani has been a practicing surgeon in head and neck oncology, a hospital manager of National Cancer Center and Tartu University Hospitals, Secretary General of the Ministry of Social Affairs of Estonia, Chief of Staff of the President of Estonia, an active member of the team that orchestrated transition of the Estonian healthcare from the Soviet type system to now what is seen as the most cost efficient healthcare system globally as per World Bank. He is amongst the initiators and architects of the Estonia Genome project. He is also an entrepreneur and a consultant in healthcare, medical technology and technology infrastructure, chairman of Tartu Biotechnology Park, and also the chairman of ScanBalt, which is the Scandinavian Baltic countries, a biotech meta cluster composed of health and bioeconomy communities in Denmark, Estonia, Finland, Iceland, Latvia, Lithuania, Norway, Poland, Sweden, northern Germany, northern Netherlands, and northwestern Russia. It covers over 60 universities and 3,000 plus companies. What a privilege it is for us to have you, Janus. It's been my personal privilege to have known you and to have interacted with you. I'm enormously impressed by the way you think, what you have done, and I hope that today you're going to share with us a little bit of that experience. So over to you, Janus.
So Janusz, this is Sachin here, and request you to share your presentation. I have made you a presenter.
Okay, thank you very much. Thank you very much, Ajit, for those kind words. Maybe it was a bit overestimated, but yes, it's my privilege to be here with you today and share some experience that we have had in Estonia on introducing concept of personalized medicine or personalized healthcare, as we tend to say. And in order to go back and start from the basics, I would like to remind what can be, how we can actually look to the healthcare or diseases or disease predisposition if we look to three main building blocks, which is genetics, or what is our bodies consist of, the inevitable process of aging, and environment and lifestyle, and which coupled with comorbidities. And if we look this, we know that we get at birth our genetic setup, which then by increasing age there will be starting changes in our body, which is actually added by influence of the environment and also very strongly lifestyle influenced. So, and finally, we can see that it can lead to some disease development. So this is, we have to keep in mind if we are looking to the actually modern concepts, and the modern concepts is that we have enormous development during last 30 or so years in genetics, in biomarker invention. Age is much more well known, and we have much more information now that we can collect, store, and process from the environmental, lifestyle, and also diseases. So, and based on this, we can say that we have today the new concept which has been emerged, which is called 3P healthcare, which means predictive, preventive, personalized, and the last is participatory. So How we have reached this is, as was said also already by Ajit, that there are now 5 pillars what we can actually rely on these new concepts. One is we have ability to analyze billions of data points and that have been generated for each individual population. We can collect them, store them, and analyze. We have a computer capacity to do this. We have new ways of molecular and cellular origin markers that actually are characterizing the symptoms of diseases. The accelerating biomedical innovation is emerging as discovery in science as integrated with disease care and wellness care. And of course, we can see that these concepts are actually going beyond the clinics to the active preservation and enhancement of wellness. What we now can see that there is a wellness industry which is beginning to emerge, and this is now brings us to the new source of economic growth. So these 4, 5 sort of factors which we can actually say will transform the healthcare sector, and we see that one of the sort of landmark articles which has been published by US team of Dr. Mauricio Flores, Gustavo Glusman, Christine Bogard, Nathan D. Price, and most important, Leroy Hood from Institute of Systems Biology. Well, they have been putting forward this 4P healthcare concept, which is, as is shown here, sort of in a simple graph, is systems biology, consumer driver healthcare, and social networks, which means actually information and digital revolution. So all this together actually been enabling factors that we can actually now step to the next sort of phase of how we see diseases and how we look to the healthcare. If we look to the bigger sort of scenery or an ecosystem, then we see that the P4 medicine is just one part of the personalized healthcare, which is actually in parallel with precision medicine, which is actually gives us a new molecular taxonomy of diseases, or stratified medicine, where we have biomarkers, diagnostics, and drugs together. This we can call this a personalized medicine, where when we add also genomics here. And finally, we have a concept of fully personalized healthcare approach. So what I want to bring you is actually what we have reached in Estonia. Estonia is a tiny country compared to India, which is a subcontinent. And if broadly we can say that Estonia is 1,000 times smaller by population than India. That's why we can also look Estonia as a sort of pilot site for the bigger countries, almost maybe even globally. We started with this already some 20 plus years ago, more actively. Here is some facts about Estonia for those who have not been so much acquainted about Estonia. So the population is 1.3 million, and the size of land area is just nearly similar like in Denmark. So what we have enabling factor, and specifically in Estonia for last 15 years or let's say 18 years, what we call the e-society, or we have a good opportunity to actually analyze, collect, and sort of transfer data securely. And what our main basis of this is actually the 9-digit personal identification system, which is a mandatory to all citizens. And today, 95% of citizens actually have active ID cards, which are chip cards, which can be used for actually authorization and identification of every citizen. Why it's important? Because this enables us huge number of public e-services, which also includes public e-health system, where people can actually be actively commanding and involved in managing their data. So one thing is you can actually use the services like e-prescription or digital signature, but you can also be in control of your data, because you as a person can always see and control who is actually have been looking to your data, who from the public service, public officials, and this gives enormous power to the actually each person. This is the next layer is we have called X-Road, which enables a secure data exchange between different data sets, which are kept in different locations and under different institutions' custody. So it means that we don't have one huge database centrally collected, which we see that some countries, even for healthcare, are trying to do. Our concept is that we have a decentralized system where we can actually easily, by using this secure data exchange platform, collect data and exchange data between different databases. For example, if doctor is starting e-prescription, then the actually system is simply collecting different pieces of data and authorization controls from different databases, and finally, which takes, of course, milliseconds, there is a e-prescription in the healthcare. So why I'm talking this, because this is a basis for the actual way how you can take real advantage to the practical healthcare of new concepts of a personalized healthcare as well. You can also use this for actually supporting disruption in the research based on health data, genomic data, and biomarkers data. So in 1999, we started a project, then what we called Estonian Genome Bank. This was initiative where we were initially aiming to collect genomic data from all 1.3 million inhabitants in Estonia. Of course, with actually facing financial realities and changing in the approach and attitudes in early 2000s, where it actually we realized that not only in Estonia but everywhere, that just to sequence the genome doesn't give you actually functions and results that can be implemented, and that the real sort of outcome is much further than 5 to 10 years. So today we are in the situation where we have 150,000 participants, which means like 15% of adult population of the whole country have been collected, have donated, we call donated, have given their sample, blood samples, which has been now stored and even genotyped with Illumina chip. And our approach is that we try to use, like we call it poor man solution, where we can actually use the data that is derived from this chip to impute it to the full sequence based on the reference genome that have been actually produced on the whole population samples. So what is important here to emphasize that as Ajit already mentioned, important is of course ethics and regulations. So we have started from the day one from a special regulation, which is called Estonian Human Genes Research Act, which was as a step one what was drafted and adopted by Estonian Parliament. And this law also foresees the broad possibility for the broad informed consent, which is also the second basis or pillar of this project, which means that like 99.9% of participants have given their consent to collect data from different databases, from clinics, from hospitals, from primary healthcare centers, but also other available data which might have relevance for the research of diseases or biological processes. And this means that we can collect data on personalized way, which can be then analyzed together with genomic data. So again, what we have here is a sample collection of approximately 150,000 participants, which is not only DNA, but also plasma. And from those 150,000, as I said, all these are genotyped, but we have also done 2,500 full genome sequences, plus 2,500 exomes. And based on this data, we have been able to produce or calculate these most common genomic allele variants that are in Estonia. This is actually, based on this, we can actually calculate back from this chip data, genotyping data, to the full sequences, which are not 100% exact, but still close to 98% what we have been collecting. So this data is actually now, as I said, combined with different time point health data collections, and this actually enables us to look for different diseases, and the data is actually collected longitudinally, so it means that we can actually see what is happening and what kind of diseases are evolving actually after the person has been joining this data set. So what is our advantages of our ecosystem? What has enabled us to do this is we have taken the research model, which is we call genotype first, which enables us to introduce and you start smart clinical trials and digital healthcare models, which means that we can look to the digital genomic data and select specific clinical trials groups of interest, and we also can actually stratify population based on their genomic variants, and then use it to actually introduce different interventions in the healthcare. So this is I will give you some examples a bit later. So then the next is what I also referred, is that we have fully connected digital health ecosystem, which means we have each person can be identified electronically, and we have several ways to actually involve people. One example is what I was 2 weeks ago in India. We had elections, general elections last Sunday, but before that we have had a 1 week period when it was possible for electronic voting over internet, and I was just making my poll sitting in Delhi in an office. So this is actually how people can be connected with not only with the government but between each other and companies, and also how they can be involved in research easily. For example, now we don't take any more paper-based informed consent, but people can go to the special website of Estonian Genome Bank, log in, just authorizing themselves with their digital ID and digital signature, and then they can actually give their consent. No need for lots of papers and lots of visits to the doctors. So, and this means that we have access to the clinical data to the national level, which enables us to collect data, but also engage stakeholders. And most probably Estonia will be by 2025 the first genetically profiled country, fully genetically profiled, because now this year another 50,000 is actually planned to add to this database. So during next years, we think that each new generation will have genotype. So by 2025 or latest by 2030, 100% of population will have genetically profiled. So why is it important? And then I think will be much more exciting just from the collection of data or theoretical approach. We can see how you can actually use this data for if we emphasize to the practical implications in the healthcare. And one way is to actually see, can you actually profile or stratify people and use this for something practical and something good for the population? Today we know that the main instrument, the main approach to actually prevent or early detection of cancers or like breast cancer is screening. We know that there are, in most developed countries, there are screening programs where the women in certain age, like starting from 50 or 55, are invited every second year, for example, for the mammography. But we know also that the efficacy and efficiency of this approach is not very high, and the sensitivity and specificity of mammography is not high either. So this means that we might have better approach, and there also we know that there are some cancer variants that are early onset and are quite malignant, and we miss them. So we have BRCA specific one or specific mutation-based disease, but there is also a notion of polygenic breast cancer susceptibility. And here it's recently published work based on Estonian research group, which actually shows that you can have in highest top 5 risk group with polygenic risk is like up to 5 times more probability to the control group to get cancer during the lifetime. So it's pretty high. And now the concept is that we could actually stratify people, women, based on the genetic, and based on this take already actions, more profound follow-up. For example, instead of mammography, rely on the MRI to do this more often, but at the same time not to border with those population groups who have genetically low risk revealed. This saves us money and hopefully will give us much better prediction and prevention of this malignant disease. So this is one example where Estonia has now started a pilot, which we try during next 2 years to implement this with primary healthcare doctor and oncologists, to actually first to look for those group of this 150,000, but also engage a new family members of people with high risk and widen this based on sort of targeted prevention programs. The other pilot what we have started is based on the one research what is done in other countries as well, like in Finland. This is a one publication from 2013 where Finnish colleagues have been screening 100,000 people on cardiovascular genetic risks. And what they found out and calculated that they followed them 10 years, and they saw that there is like 135 cases which were missed if they wouldn't actually look to the risk stratification. So here again, we are looking to the possibility to only apply, for example, statin treatment only to the high risks, upper 20% risk group, and not to treat the people who have low risk. So we think it will be economically sort of reasonable, but also we know that the statin treatment has a specific side effects, complications, and we can avoid them. So this is one approach. What two projects what we are now already actively pursuing in Estonia in the public health system, and we hope that we can actually report positive results in 2 years. This phase is actually already the practical piloting, and if this works, of course, we try to go to the other areas. Now it's also diabetes. This is is in Estonian, unfortunately, but it actually says that you can also screen a population genetically for the diabetes and give them some risk sort of predictions, personalized risk predictions, which also helps people to make decisions on the lifestyle. And if the person has overweight and also genetical predisposition, then we can actually maybe influence and motivate person to change a lifestyle, the eating habits, but more importantly maybe the physical exercise, and maybe we can return the process. So the next, which is even more important, we think, is pharmacogenetics. If we have a full database, fully genetically sort of described and profiled the population, then we can use this additionally to the current tools what we have. We are one country where we have 100% of prescriptions are actually done electronically. So it means we have database, and we on this software there is already a drug interaction tool, decision support tool, which is actually guiding doctors during the prescription process. That if there is some conflict between two drugs which have already prescribed to the person and the doctor is intending to prescribe, then there doctor can change the prescription based on this information that is provided by this decision support database. Now what we want to add is pharmacogenomics, because we know that there is approximately 1% of population where each more sort of known drug due to the CYP family genes, which are actually altering the metabolism, that can have complications, and that's why it would be better to actually screen them before we prescribe. And this type of system or environment will give us opportunity to do this. And this is now the next phase, what we are going to introduce during next 3 years. Of course, initially for those who have already actually been genotyped, but as I said, our database is widening, and by 2022, I think we will have like 300,000–400,000 already in the database, which enable us to change this current customer or client or patient pathway, if you like. So, which is described here, where we have just people will turn up to the healthcare system, either taken by ambulance or through the primary healthcare clinic, then to the hospital, then to the pharmacy, and get back to the primary healthcare. Now we can actually add other layers. We can actually prevent actively, predict and prevent actively diseases. We can actually advise people and motivate people from different genetic diseases, and also we can intervene and prevent some harmful effects and improve the efficacy of the treatment. Of course, this is what we can do in the practical medicine, but all this, as I mentioned, is open to the possibility to be used by the industrial partners for development of drugs and new diagnostic platforms. So what we are now planning also in our system is to create a commercially viable system where government can actually facilitate the actually access to the clinical and genomic data for the third partners. Today, there is quite a lengthy process because there has been restrictions which were foreseen in by the law, what I mentioned, what was the basis of the Estonian Genome Bank, but the actually understanding has been changed. In 20 years ago, when we passed the law, or it was actually 2001, then there was a lot of fears. The people were actually predicting that if somebody knows your genetic profile, then all kind of bad things can happen. The insurers can use this. Employers can use the data to discriminate. We haven't seen, and now more and more in Estonia starts to be actually prevailing attitude that genomic data are like other delicate personal health data, and you can use them in the complex with other. But of course, they have some predicting power, which you can actually see if you have tools for actually using this in reasonable way. So that's why we are now, most emphasis is actually look how you can actually consolidate this and to look to the political approach and population expectations and commercial expectations to use this wealth what we have created for the further research in healthcare and pharmacology, pharmacy. Sorry. So this was presentation from my side, and I'm happy to take questions.
Thank you. Thank you, Janis. That was very, very informative. What's also interesting is that a number of the questions that were coming up have already been answered in your this thing. So that just makes my life a little easier. We have got roughly 10 minutes and a little more maybe. Let me start by shooting some of these questions quickly. Beatriz Noya Marino, I hope I got that pronunciation right, from Spain, says something, and it's not just specifically in Spain or in the Atlantic region, but there is a big gap between research and its transformation to commercialization. Right? That's everywhere because of the long process and regulatory regimes. How will these new studies and use of data and genetics help us to speed up that process?
Well, of course, this is absolutely correct that this gap is existing everywhere. Maybe in some countries this is less, maybe US, this is more viable ecosystems, whereas there is more investment funds, investment money is available. We know that this is also has been a lot of under discussions also in Scandinavian countries, even in Sweden, what we consider one of the leading regions or countries for the taking new inventions to commercialization. We see that even Sweden, we hear about the problems where there is quite good basic research, but still not as high intake by commercial development as it might be. So what I see here, that this approach of open, of the availability and ease to get access to data might make it much more easier and facilitate actually to bring the hurdle for the inventions time-wise and investment-wise lower, because you can, and the other thing is you can start sort of hypothesis-based research. So you don't have to make an hypothesis, you have and then collect data based on your design the study and then collect, recruit people. You can go and actually to the databases and you can do in vitro and in silico research just to check based on data and health available genomic biomarker and health data how sort of viable your hypothesis is, and then if it's more sort of probable and plausible that this has some kind of novelty in and some kind of basis in your hypothesis, then you can go and do the specific research to actually confirm this and validate this. This is what I can say, that yes, it will actually somehow democratize the whole research ecosystem.
Okay, lovely. There was a few mothers, and I'm going to mix them up so one of the things that obviously startups, and I'm sure there are many young people listening to this and itching to get their hands on the data, right? So have you already evolved policies on how startups can access the data? Have you already done things on how do you mask the data so that individual privacy is protected while there is adequately detailed information for researchers to do research?
Yeah, this is, I mean, the data security has been the utmost concern from day one in our system. And this means that I can bring you an example which is even too restrictive. Today, there is a system was from the the point where data or the genomic sample with the personal data actually enters to the genome bank, immediately the personal data and identification data and the sample, physical sample, are separated, and there is actually the sample and has been given a unique code, which is kept in a special data storage, which doesn't have actually internet connection, which makes it quite troublesome. Actually, it's a lot of handwork to actually go anonymize and or pseudo-anonymize and then personalize the data back, because if you want to connect with the clinical data, then you have to know actually to which person's genome you connect. But of course, for the research purposes, initially, if there is no specific informed consent taken and permission from the ethical committee, you can only use for research, and specifically in commercial use, research, you can use only pseudo-anonymized data. There is no sort of personal data what you give out. So the other question is that all participants in this genome bank have given also informed consent that they can be recontacted. So if there is, as I brought in, if, for example, a company wants to conduct clinical trial on a specific group of people with specific genomic variants, then they can turn to the genome bank, ask ethics committee permission with the genome bank, and then genome bank can actually connect these people with a request to join the clinical trial. So of course you can select the clinical based on genomic, and also with existing information on the health status or diseases. So you can select the group and go and directly target to those people, so which make it much easier to recruit the sample size what you need. Okay. So, but yes, we have to work on this to make it more accessible and more automatized, and this is still need to be done in Estonia, for example.
So Pooja Dubey had a interesting question, which I guess is more relevant to India than it's to you, that we have large pockets of people who are not necessarily properly educated and do not fully understand the meaning of informed consent. For situations like this, how do you deal with it? I mean, obviously getting an electronic informed consent is out of the way. Even asking them to sign on a piece of paper may not make sense. So do you have mechanisms for social workers or government people to explain to them the implications of this? The flip side of that also is yet another thing, and you mentioned this in passing. People had worries about what will happen if I give my genetic information. Will it damage my genes, for example? Some of the kind of fears that people had. What did you do to overcome those fears?
So what we did is a extensive public education. This is in Estonia. This is only way. Of course, people initially who were joining, there was no electronic informed consent, but it was actually process in the first years where family doctors or GPs were actually recruiting people. They were having extensive health questionnaire-based interview, which was added the information from the primary healthcare doctors database, the health information. And during, of course, to start this, there was counseling. Today, I think in Estonia, we don't mostly have this problem, because most people have heard, they know about the genome bank, and they know more maybe about genomics than average population in other countries, because it has been extensively covered. What is important, of course, is that what happens if there is detected some risks, how to convey this, how to explain this. This is a problem, and this is what we actually emphasizing during this pilot project that I actually presented here on breast cancer, cardiovascular. How this professionals, so who should explain the person about the risks, because and what it means to have a risk, what means risks that you have 2 times higher risk than the average population, what actually means. And this is a part where we have actually having a special activities under this pilot project to educate doctors, but also we have plans to start to educate the special genomic counselors, which are not doctors, but which with like several less than the bachelor level specialists that can actually take burden from doctors to actually advise people about the effects and meaning of the results of genetics testing and susceptibility risks. But this is a very good question, and I don't have really good recipe for the large population like India. But if you divide this population to the smaller communities, then you might have some ways to solve this, because I don't think you can do it from the central government in India. It has to go down, not even to the state level. It's intended to community level. So, but it's a lot of work, of course. But from other hand, of course, it might give you rewards if you look today. I think in Estonia, the genotype cost is like one genotype is like 50 euros. If you go to the even bigger sort of scale, like in India, I think it can be much cheaper, and it might be cost efficient in long run. But of course, to start this, you need funds to do this, and maybe some people are able and willing to take this investment by themselves. We know that there's has been a project in Switzerland which called Health Bank, which was meant to be like a cooperative where people will join and pay for the genotyping, and themselves giving also permission to use their data for research.
Yeah, Janusz, just intervening here. Yeah, so Mr. Ajit and Janusz, I just want to intervene here that we probably can take one large question, because I was reading there are probably a number of questions and we don't have the time. So this is a question to both of you, and I'm acting as a, let's say, a voice of the audience here, just to summarize this for you. So that there is one part which is the Indian part. So we heard the, let's say, something like a science fiction from our point of view, from the Indian point of view, Mr. Ajit. How do we bring this future to India? What are we? What is your observation in terms of the regulatory framework? I think there are a number of questions people are asking, and you know the challenges and the opportunities. How do we leapfrog? And second question to you, Janusz, which I think some people are asking, is what also Dr. Ajit highlighted in the beginning, that we are a very diverse country in terms of the genetic pool. Is Estonia very homogeneous? Is that a factor for you to predict, let's say, what you showed in your presentation? Or do you foresee that Indian population can also benefit in spite of being diverse? So over to both of you. You can take this as a last question before we wrap up.
Okay. So let me hit off the Indian this thing. The Indian government is working on policy, and I think the public consultations will probably begin. There are a number of working groups working on it. The government has recognized that this needs to be done. A number of national research institutions are actively working in some of these issues from the research point of view. I think there are some of us who are, and certainly we are doing that in Hyderabad, trying to see how we create that accumulation or or a federation of all various players, from the clinicians to the researchers to the corporates to the startups and so on, on this whole issue of genome-based research. Now, a lot of that is also going to mean we need biobanks, and that's again a big initiative that has been started once again here in Hyderabad. I'm not just trying to tout Hyderabad; it's partly because I know more about Hyderabad, but I think we need to do a lot more, and we need to collaborate with people like Janus, because they have developed standards, they have understood what this is, and we need to figure out how to adapt it to our specific requirements. We need working groups to do that. It's happening. This big strength that I see we have is, as we get into more precision and personalized medicine, even the multinational companies or anybody who wants to sell will need data to tailor it to that. And if we as a nation can create an access to our data, if we create a massive data movement of a national biobank, for example, that could allow us to be partners in this entire future, discovery of medicine. And that's why I was asking Janus questions about how do we ensure that that data is protected, how do we ensure that that data is available to researchers and to people like that. But I think obviously a close collaboration with Janus and his team and their advice is going to be crucial for us to get started on the right note. Over to you, Janus.
Thank you. So, if we look to this heterogeneity, yes, Estonia is obviously, compared to India, homogeneous population, because of just, if you take a size of Estonia, it is 1,000 times less, and of course there are some extremes, but basically it has been developing steadily due to the mixing different ethnic groups. One of my friends used to say that Estonia has been in the crossroads during the history, and all kinds of different nations have been downloading the DNA here. So we can say we have done research, and Estonian population is pretty similar to the rest of the population in the Northern Europe. Maybe if you look to Finns, then the Finnish population is even more diverse or separate than the common sort of genomic profile in our region. So how to approach in India? Difficult to say, but again, as I said, I think India shouldn't be taken as one country. It has to be differentiated to the different region, different groups, and then if you look at the smaller subsets, then you might see that these smaller subsets are pretty homogeneous, because I don't think people have a lot of sort of migration in India. I don't know, of course, I might be mistaken, but still, if you go to the small enough subsets, and 1 million in India is very, very small subset, then you might see that this population is quite homogeneous, and you can actually maybe look at the bigger region. But of course, you need a research on this. It's just my guess at this point. And of course, there might be a lot of information available. I'm not aware of. I'm not been looking at this specifically.
So jumping here again, if you both permit me, as you know, we are now exceeding our time, but at the same time, you both might agree that this is a topic which probably requires us a longer conversation. And if I have your permission, maybe we can identify subtopics within this larger topic, and maybe with the leadership of Rich, we can see, how can we do more constructive discussion in this direction, and maybe, out of this webinar, actually build some actual collaboration. So maybe, Mr. Ajit, if you can give some last words, that what is the way forward here? Should we take some action?
So we have already started taking action, and in fact, on the sidelines of BioAsia, Janus and I had a very brief discussion, and we are hoping to take that to a more formal collaboration between him and also, of course, the entire Scandinavian Baltic region, so that India can become a partner, at least in terms of how do we follow common protocols, common data, common basis, and so on, and what do we learn from their experience. So yes, certainly this is a big area. We also need to understand how they keep biobanks, how they protect data. So there's a large variety of things on which we need to collaborate with them.
Right. So again, I would like to thank both of you for taking out your valuable time and creating this conversation. As you know, Innovation Curious, our interest is to bridge innovation in the health sector, and Rich is our ecosystem partner. So we're really grateful to your partnership to to make this conversation possible. And for this webinar series, I especially want to thank ScanBalt Forum, which is one of the organizing partner, along with Tartu Biotech Park. And this was the third in this series of our webinar that we did with ScanBalt and Tartu Biotech Park. I request our listeners that they may share with us on the email mentioned if there are any topics of interest that we can again reach out to our collaborators like Rich and Skanbal to organize further sessions on these topics. Thank you again. Have a good day.
Thank you, Sachin.
Thank you.
Thank you, Jan.
Thank you very much. Thank you. Take care.
Bye.